Very good. Okay, so good afternoon, everyone, and welcome to the Theoretical and Computational Biophysics Seminar Series. We are hosting Dr. Bernardi from Auburn University. Rafael doesn't need any introduction to all of us, actually, but just for people who might be on Zoom, I'm going to introduce you briefly. So Rafael received his physics degrees, bachelor's and master's in science in physics from Brazil. And then he did actually a PhD, BSc, Doctor of Science at the University of Biophysics, also in Rio de Janeiro, before spending some time as a computational chemistry researcher at UPenn. And then he did a postdoctoral research associate position here with Klaus Schulten for 3 years, 2017, 4 years. 4 years, okay. So, and he became actually really a critical part of the group in many respects. So he really participated in many activities of the group. He was really an essential part of the center for a long time. So Rafael is an expert in mechanobiology, as you will see from his talk actually today. But in addition to that, he has been also a main developer of features who are NAMD and VMD. So he was the main developer of the QMMM code that made with San and Klaus. QuakeMD, that many of you know, was essentially Rafael and his friends. And now actually he is implemented AlphaFold in VMD, a plugin for that done by Rafael's postdoc, and he continues to be one of the contributors to our software packages of the center. So with that, thank you very much. Rafael just happened to be around in the neighborhood, so we asked him to give us a seminar and update us on his research. It's great to have you here, Rafael, and we look forward to your seminar.
Thank you very much for the kind words, and thank you for inviting me here. It's very nice to be talking down. I'm going to talk to you today about some of the things that we are doing with some of the projects we were actually doing before, but like we have been using some new techniques on it. So before I start on so what I've been doing there is basically the same thing I was doing here. So like my group there works on some method development. So we are doing some molecular dynamics methods. We're working on that, and also some artificial intelligence for analysis of the method for the trajectories. And particularly interested in mechanobiology. So applying this to mechanobiology. So the mechanics of proteins, mechanical stability, and also the evolution of force resistance. So this is the physics building where we are there. And our lab is actually like in the opposite corner, like this part. Why we care about the mechanics of proteins, why I care about that is like the mechanics of proteins regulate a lot of different aspects of the life of a cell. So if you have some mechanical signal, that mechanical signal can be translated into a biochemical signal that can be transduced into something that the cell is doing. Or you can also have the opposite. You can have some of these mechanical signals, some of these biochemical signals that make at the force stroke, so you make the cell act and using motors to do that. But also, if you look outside of the cells, mechanoproteins are responsible for adhesion, like one cell to another, to like bacteria when they interact with us, or with the virus, like we're talking about coronavirus these days, and like when the coronavirus is all mechanical proteins. So like we're interested in all those aspects. Okay. How do we measure the force? We're interested in the mechanics of proteins, we're interested in force. How do we measure that? Experimentally, that is done mostly by using one of these methods. So either using tweezers, you can use magnetic tweezers by having magnetic bead, then you can pull the protein of your interest. Basically, like when you move this magnetic field, you can apply a force in that bead. So you can do the same thing with optical tweezers. You're going to have an optical trap, you're going to trap some molecule in that optical trap, and then you're going to move that light, and so by doing that, you actually apply force on the system. Or you can use atomic force microscopes that are more frequently seen this kind of problems. Now you're seeing more of the others, but like in the beginning, particularly, you're seeing more of the atomic force microscope. What you do is you have a protein attached to the cantilever of the microscope. The other side of the protein attached to a surface, and when you move this cantilever up and down, you actually get a signal for the force, how the system is behaving. Another way that's more recent is to use a centrifugal force microscope. And what you do there is you have basically the same idea of the optical freezer. You're going to have a sphere that you can detect with a fluorescent microscope. But instead of using a optical trap, you have the molecules attached to a surface. And then when you spin very fast, you're going to have a centrifugal force, right? Like, so you're going to basically make all these molecules go away from the glass that is in the center. And by doing that, you can track all these spheres at the same time. So you actually do a single model experiment, but in bulk. So you do many of them at the same time. So the advantage for this method is that you can actually do the same thing that you do in AFM, but instead of doing one at a time, that takes like a whole day to get 1,000 traces, this one takes minutes to take 1,000 traces. So it's much faster. Which is important if you have something that degrades fast or something that is not so stable. When we do that computationally, what we're doing is something similar to what is done in the AFM. So mostly we have been working with Michael Nash and Herman Dahl to do the AFM experiment. And what we do in the computer is basically simulate the same thing. You use thermodynamics, you have a spring that you use Hooke's law and you're pulling on the system. And by doing that, you can calculate, you can check how is the force extension curve, the same way you would get in the AFM. The difference is now in the AFM, you only have the force extension curve. If you want to test anything, if you have any hypothesis, you need to go back to the bench, make a new molecule, see if that molecule now behaves the way you expected by that thing that you believe was happening, and do it again. With the computer, you can analyze the trajectory and see why you have the feature, or you have an extra peak or something like that. So what we've been doing with SMD, I've been calling it in-silico single molecular spectroscopy. You're using the same principle, you use the same thing that you're used to. I don't need to explain any of that to you. And basically, you're adding this extra term when you're calculating the forces, like I'm saying, the same principle that we had. But we do that hundreds of times with about 300,000 atoms every time. We are using MD and the things that you're all used to. And we're seeing how these molecules are moving all the time, trying to analyze all these trajectories, see what is happening at the same time in all the systems. But the most important that we have been doing that is different from any of the papers that you see in the area is that when you look at most people, they are doing 3, 5 replicas. We have been trying to do thousands. So what we do is, this is a paper that we published before, is what we have been calling like wide sampling. It's not any enhanced sampling, it's just like doing a bunch of simulations. And what we do is that doing all the simulations, we can actually try to use the same metrics that we use in experiments to analyze our simulation data. Different from enhanced sampling, where you're trying to like better sample your free energy landscape. You're not doing that, you just want to get the same type of replica. And that's important because if you look, let's imagine you do simulations and you do just like once. Okay, all these colorful points here are from simulation. If you get one time simulation, you might be measuring at this point here, or maybe 2 or 3 points around here, and then the next time you're measuring these 2 or 3 points here. So these are different conditions. You might get the wrong slope. You might get a different interpretation of the result. So to get a distribution like this, the minimum you need is something like 30 replicas. If you get hundreds, you get more beautiful distributions. And so you start to get the distribution that can actually fit the experimental data. So you're fitting experimental data without actually needing to fit it. Like you're getting to the same point without fitting it. Okay, that's not so simple to do, especially in the past. So of course, we're taking advantage of what's happening in the world. You know, like there's like these exascale computers coming up, and not that you're using exascale computers for that, but we've got this advantage that petascale computer that was the revolution when I started in Paul's lab that helped, you know, like to get the structure of HIV and things like that are becoming cheaper. So basically, you can have a petascale computer in your office now. That's too hot probably, but like you can have it in your group. And by getting something like that, you actually can do much more than you could do in the past. And in our group, we have 3 of those, like this is the rack we have, and they're integrated. And just to give you an idea, the way they're using it, that's basically forcing a bunch of simulations to run in parallel at the same time. We don't just run one job per GPU. We run like 10 jobs per GPU at the same time. We're getting the same efficiency that we could get with 400 nodes of blue waters per node. So that's actually, like, it's basically I have my own allocation of blue waters without applying for any computer time. And that made us very successful in getting like very fast results in the last year. So
So, sorry, may I interrupt you? You said multiple jobs on the single GPU,
As long as we have memory, we keep adding down in the same view.
and this is just for them to backfill each other.
Exactly.
Okay, I see, I see.
So what happened is like you're basically using all this, the time that, if an MG or any code was perfect, you would not need to do that.
Right,
But because no code is perfect,
right,
you're actually using this, like there's never like a time that the machine is not required.
I see,
It's always at 100%.
I see. Gotcha, thanks.
So
Mhm.
we actually, like when we were doing some of the simulations for the COVID, in just over 2 or 3 months, we were able, we got like 200 terabytes of data. So like it's so fast we were running this.
Mhm.
So, but like, what can we learn with this? Okay, we are using these ideas. We are basically doing many replicas. What can we learn with that? So when I started here, when I started studying the systems, this is what we knew about strong proteins. So if you have like a channel or most things that are inside the cells, they would break. Okay, this is the force that you need to open or doing some movement or breaking something. Experimental measurements. Okay, so like if you have something like inside a cell in less than 100 piconewtons, like most systems that you know, they actually act here. You know, like 1 piconewton, 2 piconewton, less than that. If you have streptavidin-biotin, which is relatively strong, will be anything between 100 and 400. There are some reasons for this big range. And covalent bonds were down here in the 2,000, 4,000, 5,000, like the only known in the region. So we were able to find other systems that are actually stronger, like the cellulosomes. We were able to find more cellulosomes, and we were able to find bacterial adhesins that are actually much stronger to the same level of the covalent bonds. And that's actually the main thing that we are studying also now. We continue to study them because we actually found some very cool things about it. Before I go into that, just because there's a pandemic going on, you might be wondering what happens to COVID. It's actually in the same regime here. But there are some very interesting things that we developed for COVID that actually are using now for the other, for the bacterial adhesins. And we're getting some very interesting results from that. But before I continue, I just want to say that none of that would be possible without these wonderful collaborators that we have. Everything that we're going to show here that has also been done experimentally by either Herman or Michael or Jan. So Herman is in Munich and Michael is in Basel, and Jan just moved to the Netherlands, to Delft.
Mhm.
So what we look at in the COVID case, like we were looking at the interface between ACE2 and the spike. You all know about it, but there are many reasons why you want to study that. This is the size of the virus. But when you go inside, when you go and look at the interface between the spike and the human receptor, ACE2, you're going to start to look, okay, the part that actually interacts there, the part that might have some mechanical properties, the interaction between RBD and ACE2. Let's look at the properties of that. To do that, we're going to look at the crystal of that system. So only looking at this part of the system, this is the interaction that they make there, like the interaction with the cell that you're having inside. Experimentally, what we needed to do was, it's too complicated to measure the system. It's weak, like a lot of complicated aspects of it. So the way we did it was actually to create a linker connecting the RBD to the ACE2. So it now became one protein. So when you're doing single molecule, you're actually doing single molecule.
Mhm.
You're measuring every time the same molecule. So you go up and down, it's actually always measuring the same molecule. And then we did the same thing with the AFM and the magnetic tweezer. The cool thing about it is that magnetic tweezer operates in a very low force regime. It pulls the protein, holds it there, and you can see how long the bond leaves when you're doing that. While in the AFM, we pull at a constant velocity, and we see how strong is the bond if you keep pulling. Okay, so you're measuring actually two different things. But first of all, when we did that, they were not sure. The results that they got was something like this. These multiple peaks, and they had no idea why there were these multiple peaks, that we could not be sure where these peaks were coming from. When we decided, okay, we're going to need to do the simulation the same way with this longer linker, like this long linker. So when we did that, we actually observed that the long linker is actually the unfolding of the RBD. Okay, so like when you're actually pulling on that system that way, you actually make the RBD unfold. So all these peaks are actually because of the RBD unfolding. Then, if you look at the first peak in this simulation here, where we are having the unfolding happen, and the one that we don't have, where we don't have the linker, you actually see that it's basically the same idea. They hit the same peak. So we don't need to simulate this longer linker. It reproduces the same thing. It's based, the linker is not actually influencing the system. The linker is just there, staying there. So there's no problem actually using it. Okay, so the first two results we got about this, it's actually kind of hidden here. The green plots that you're seeing here, the blue plot, the difference between the two is that for the green one, you're looking at SARS-CoV-2. The blue one, you're looking at SARS-CoV-1. So actually, over the years, SARS-CoV-2, the coronavirus became stronger. Like it's harder to break the complex of SARS-CoV-2 than it is to break the complex of SARS-CoV-1. It's actually more than twofold the difference. Okay, which seems amazing. But is that important for the virulence? We don't know, but that could be one of the points. So then we started to analyze how that happens using cross correlation, like, and I think Zen knows a lot about that. So using network analysis and all these systems. And what we did then was actually, can we use network analysis to generate a new molecule that is not SARS-CoV-1, that is not SARS-CoV-2, but is something in between? So we compare SARS-CoV-1 to SARS-CoV-2, calculated across the delta of the correlation, so checking what are the differences between the two. And then we found that there are some regions that were more like highlighted in that direction. And if we look at these regions, we could maybe create a SARS-CoV-1 with a few mutations that would actually reproduce the properties of SARS-CoV-2. And that's what we did. And by selecting 8 residues, we were able to make a SARS-CoV peak force. Now, you don't need to look at the distribution because that depends on the things, but if you look just at the peak force, this is SARS-CoV-1, this is SARS-CoV-2, we were able to get a chimera here that we call a chimera. So it's a mutated SARS-CoV-1 that actually gets very similar to the strength of the SARS-CoV-2 with only a few mutations. There are hundreds of mutations. You only change a few of them, you're able to recover the force. So to make the force, just a few of them are important. Okay, so that's some of the results we got here. We have another paper that is submitted for analyzing all the variants using the same ideas. But I think that the main thing I want to show you here is that, okay, we were able to do that. We were able to confirm that experimentally. Now, if you look at this, can we use the same ideas to study staph infections? That is something that I've been looking at.
So before you go, we are still looking at RBD spike interaction.
Exactly.
In the correlation.
I see. It's the same thing. It's just that we started to mutate the RBD only in a few places, and the positions were selected based on the network. It's not on hydrogen bonds or anything like that, but instead of which bonds are stronger in the SARS-CoV-2, more correlated in the SARS-CoV-2 than they are in the SARS-CoV-1. And we mutated only the RBD. We kept the same H2.
I see.
So Rafi, did you look at the other variations of Omicron that have so many more variations?
Yes, we have all of that too.
Yeah, so.
It's not here, but yeah, we have actually.
Oh, that's in your next paper? Or-
Yeah, that's in the.
Have you done the same thing for the antibodies binding to the virus?
We didn't turn the antibodies, but it's something that we think about it, yes. We were thinking about it, but we never actually did. We actually, because of the results we have, we submitted a proposal for the NIH to study, can we design these things actually for real? Can we actually use this to design
Better.
antibodies, basically.
Yeah.
It's not a traditional antibody, but can we design antibodies? And we got a good start, but was not funded. We just resubmitted, just resubmitted Friday. So the plan is actually to try to do that. The extra step that we are doing there is use some AI to predict, because we got to a point that it's just too many. We're actually using way too many variants. And that's actually, we are doing the same thing for staph. So it's the same idea. And I can show you.
Are those mutations only at the binding interface?
They're very close to the binding interface. Yeah. So they're either a direct contact or the first layer. Maybe they're stabilizing a direct contact.
So was there anything that wasn't on the interface, but was still interesting?
There are some points that are not in the interface, but you're seeing more that now looking at another aspect of it, that is high force versus low force binding. So what happened with thermal binding versus what happened in the cooling situation. That we are seeing some points that are farther away.
Yeah, cool.
But okay, then going back to the staph infection, so we we want to look at that. And the reason why we're looking at that, it was an obvious one, right? So about 65% of all bacterial infections that happen are from staph. They are the most deadly one, typically. Staphylococcus aureus, Staphylococcus pyramidis are the main ones that you get. And when you, one of the main virulent factors for them are the adherence staph. So like how actually they start the infection is you have some bacteria, they start to attach to a part of your cell that or your body that they are building this biofilm on. They start to grow, they get to maturity where they have a biofilm, then some parts start to get out and start to invade other parts of your system. Depending on where you are, where this is starting, you're going to have a different range of forces that this thing is going to be subjected to. So, for instance, the urinary tract is a lot. If it is in your lungs, it's also a lot. If it is in capillarities, like very small parts, like very small veins of your body, almost nothing. So, like it really depends on where you are. You're going to have different mechanisms, a different like you'll be subjected to different forces, and then we're having different mechanisms for environment. So, what are the mechanisms that we see for force resistance? So if you look at that again, we're looking at this step attachment. We're going to look at how these proteins actually interact with the human extracellular matrix. And this is from the Staphylococci aureus. The way it works, you have something like this. You have this protein from the bacteria that have two domains there, the N2 and the N3 domain. The specific protein that we looked at before is the SBRG. So it interacts with the fibrinogen coding of your cells. It starts with a peptide at the tip of the C-terminal of this protein, that is of the fibrinogen coding. And when you pull them apart, basically, like you can rupture this in different ways. Well, you can have some thermal filtration that's just going to go away, or you can actually make it very strong. We showed that before. To measure that, what we do is in the AFM, you hold one end in the other. We do the same thing with the simulation. So you hold in one end, pull in the other. All these colorful parts here, we're going to look, but basically you have the peptide in orange, and the blue and green part are the same protein, the same molecule, but the green part is what we call the latch that closes on top of the peptide when it binds. So it's open in general, like if you crystallize it like in paper form, the end should are flexible. When you put the peptide together, closes the latch, and this thing becomes very rigid. Okay, so like all this green part is very flexible without this orange part. When we pull that system, that's something that we did a few years ago. We end up getting these extreme high forces, like both in the experiments on the left-hand side and in the simulation on the right-hand side. When we did that, we started to look at the plot that you saw before. You want to get all these simulations, and simulations can reproduce the data. Like fitting using like the fitting he's using from the Zabu model. But one interesting thing is like that you can look at it right away is that some people talk about it for many years that they still have a huge gap between the two. There's this 5 orders of magnitude gap in speed between the two. And this comes from one of the things in terms of software development that I know this group is interested in. That is, when we do that with other methods like Martini. The Martini tree is so much better than the other parse grain methods. So can we actually use it? And the answer in principle is no, because these are the results from parse grain using Martini and Gromax. So we get a lot of data. And you see like the forces are actually much, much lower because interactions are not so well determined. Like this interactions between two domains. But we didn't give up on looking at that, because if you look at this, we still have the same idea of the same curve potential, same kind of. Can we actually do something about it? Can we reparameterize Martini in a way, not Martini itself? And that's what we got at the end. So what we did was the following. We went to the smallest speed that we could do with the simulations, like with our atom simulations. And we compared the same speed with the coarse grain. And then we just assumed that that was a perfect case where they would match. And then we used that number to multiply to the Martini. Okay, so like the forces in that range are 3 times higher. So let's multiply by 3. The loading rate is missing by 8 times. Let's multiply by 8. And then when you do that, you end up just using one of the points, should be, which is, I think, this one here. And then when you do that, you end up multiplying the same thing for all the others. and you get this curve back to the place it should be. So like we're calculating how much is off. We did the same thing for another system, it worked as well. So far it's kind of working.
With the same fudge factor? With
Yeah, the same factor that you find for one system is going to work well, always the same system.
Yes.
It's per system. Basically, it's the fudge factor for a system. If you use the same factor for the same system, it seems to work.
Okay. For the same system.
For the same system. Which is at least giving you some idea about what is happening there. But Martin is always weaker. The interactions are always weaker in Martin. And so that at least is giving us some like, can we close this gap using some ideas like, and that's something that we're trying. Okay. That's one of the things we are actually trying to run nowadays at 1 order and 2 orders of magnitude lower. We're first going to see if we can actually get the experimental result of getting off of that curve in the same. Okay, so that's one of the things we're trying. Then moving on, we want to study how this system is behaving in the two situations, the AFM and the tweezer, because we want to see how the system actually behaves both. The problem you have experimentally is that this thing leaves too long in the tweezer. So when you try to pull this system with a tweezer, you're holding it in force and then you need to keep it holding the system. And after a few hours, it's still there. It does not break. So that makes things complicated because it's like a catch ball behavior. So the way we can analyze what is happening here is they have 2 different unbinding pathways. We came up with a new idea that is basically, okay, what you're doing in the trees is basically holding the system, and then you're gonna let the system unfold, and then let the system unbind. Okay, we cannot unbind the simulation. This is not take hours, right? So we cannot do it, like hours of real time. So we decided to just analyze what happens when you're doing it. That's very long simulations, many replicas, and analyze what happens with the interfaces. And do the same thing for when you're pulling. So you can already see that when you're pulling, things are much more stable because you're actually constraining it much more. But the result that we see is that when you are pulling the system, if you calculate if you use network analysis to calculate the force propagation pathway, you're going to find one pathway of low force dissociation, one pathway for high force dissociation. Curiously enough, the low force dissociation pathway goes directly to the latch, going to the peptide, and go kind of straight. And the high force one diverges around, it does not go to the latch. Okay? So the principle that we are trying to look at something that we believe, and it's something that we have experimental evidence, that there are 2 different pathways, one of high force dissociation, one of low force, which makes a lot of sense. You go from an unbound to the bound. Imagine the bound from the bound from bound, sorry. Imagine the following. If you're trying to remove something, like let's say you're trying to remove this from my hand and try to pull it to the side, what is going to happen is like it's hard, it's hard to do this. So, or maybe it's easier because you're going to open your hands. Like it's a different mechanism than doing this. So these are 2 different pathways, 2 different mechanisms. That's basically what you're doing. So can we actually measure that? Can we actually get a metric for that? So then Marcelo came up with a cool idea, you also know him, right? So Marcelo came up with a cool idea that is like, can we actually compare the correlation difference between the system, like between the simulation where we're pulling and the simulation that we are not pulling, just holding, and then calculate what is the correlation difference between the 2. Okay? So these are like all the residues from the peptide and all the residues from the protein interacting with the residue. And when you see these big spheres here, it's because, like in blue, it's because the correlation is much larger in the one that we're pulling. When it's orange, it's larger on the one that we are just holding. And if you look at this, in basically all of them is kind of going to the blue, right? So when you pull, the correlation actually gets higher. So there's more correlation when you pull. Okay. Now, if you look at the same thing, the comparison with the latch, so we're looking now between the green part and the blue, we see the same thing. So the latch starts to interact more with the other part within the tube as you pull. So that part becomes more rigid. Okay? So when we look at that with a Pca analysis, what we notice is that as we pull the system, what you see is that these 2 parts here, so the green and the blue, actually started to kind of smash into each other. Like they start to get like stronger and stronger contact, and they actually get much like closer to each other. And by doing that, what you have is at the end is something that is sliding versus spilling. In the low force situation, basically what you have is a thermal dissociation but restrained. So you change the rate by restraining the 2 points. You're changing the rate. You're making it live longer, but you still have the same thermal dissociation pathway. Like you still let the system open the latch, the molecule goes away. While when you're pulling it very strong, you don't allow for that pathway to exist anymore. You need to slide the peptide up. And we showed it before, the paper published in Science, that when you slide it up, you have to break like 18 hydrogen bonds at once, which is very strong. So like it's very clear to us now that these are like completely different pathways, and we can actually show it. And there are experimental evidence of what we're showing. So we have all of that too. But okay, then might come a question of, can we trust network analysis to do all these assumptions we're doing? Because we're basically having all this information from the trajectory, like thousands of atoms moving, making many replicas of simulation, and then we're reducing everything back to just a couple of contacts. Can we actually reduce that? So Marcel and I were talking about it, and then he told me like one way we can test that, and that was in my opinion, a neat idea, is if we assume that the network analysis is our training set for an AI algorithm, and we're going to use that training set to predict what high forces are going to be in other projections that we're not looking in the training set, that should give us back the force that we have for rupture. And that gives us back means that that's enough as a descriptor for the system. And that's what we did. So this is the correlation to the two. In the line would be perfect, right? Like between the training and the result. So these are the predicted rupture forces. They're all in these points here, and these are what they should be. Yeah, and this is the rupture force that we actually got for those systems that we did not use in the training set. So like what was the simulation telling us? And when we see this is reasonably good. So like it can predict for you, and it's giving you like different colors here means different velocity we're pulling, like faster pullings. And it's giving us basically the same what we were expecting. And it's better than that. This is what the distribution looks like from the predicted model that looks somewhat like what we expect from the experiments. And this is the dashed line here. The black one is the predicted model versus what we actually have in the simulation. So this is basically telling us that if you're running an SMD and we forget to save the file of the force, we can actually recover that force, without doing it. But it's better actually, because we don't actually never need to get, if we get to that point, we might never need to get to the point where we rupture the system. Because this is analyzing not the rupture point, we're analyzing before. So you start pulling the system and just stop analyzing just that part. We analyze the ramp part. So you don't actually need to continue your simulation. Maybe you can simulate only 20% of the simulation out, because you're going to assume you're going to get the force what it is later.
So from your network analysis, you put in all the correlations into your engine, into your model?
No, just a few contacts.
Just a few contacts.
Yeah, from this map here. From this one here. Just basically something.
Oh, I see.
Actually, it's from this one because it's the contact with the peptide on this. So we want to calculate basically when we lose this contact. And then we tell what the force is, which one of the simulation, and then we try to get it. So in our training set, we had like 150 simulations, and then we confirm it on the training.
But some of them have different mechanisms, you said.
Oh yeah, but we're looking only at the high force. So at each group, we analyze independently.
Gotcha.
We analyze each group independently. That's why we get different colors, the different velocities. But then there is another part of the story, because I'm telling you a lot of stories all at once. Okay, but like where all these force resilience come from? And we're talking the SARS-CoV-1 to SARS-CoV-2, that you see that the force went up over the years. Does the same thing happen with these bacterial systems? And so we started to look at them. Okay, that comes to the part where we use AlphaFold a lot. Can we look at the whole adhesion domain superfamily and look at all of them? Try to find as many as we can. So we start to look at all of the M strands from Staphylococci aureus. What we did then, we selected 200 adhesins from Staphylococci aureus. We annotated some scores, binding partners, found the peptides, put all of that into, we broke the system into the N2 and 3 domains because these are longer domains, and we took the parts we don't care about off. Then we use AlphaFold using the AlphaFold multimer, using our interface in VMD in a batch mode. So like something that some of you might know, like we created this interface for VMD. It has two modes. The one you just go there and click one molecule at a time, or you have a batch mode if you have a bunch of sequences that you want to do all together. And that's when you have 200, that's very useful. So we did that, and then we selected system from the AlphaFold because not all the systems were good. We selected 48 full-length sequences, did SMD MD in all of them. We did 29 of them. The complex was stable. It was not going away after the MD. So we kept only of those. And other than this 29, 3 of them were also crystal that we also added. We're not from the AlphaFold. So we are actually looking at 32 different systems.
So Rafi, let me be sure I understood you. So you did each domain independently, N2 and N3.
N2 and N3 not together. But we just broke apart because there is N1, N2, N3, B1, B2, B3. We just cut off just the part of the care. Just the N2 and N3, because that's the part that is the interaction between the peptide.
Oh, okay.
But
how did you use the AlphaFold multimer?
Because the peptide binds to the N2 and N3 domain. There's a peptide target, right? So in your body.
Okay.
So that's the human peptide.
And when you use this now through VMD, what do you do with the enormous database that comes with AlphaFold?
So it also has all the analysis together. You can, oh, you mean the one that you used to make the job? Or?
Yeah, because that thing is.
Yeah, we have that in our computer. We install it. We install the pool before. That's the way we do it.
Okay, but then when you use VMD, does that make use of that full database?
Yeah, you need to have that. You need to have AlphaFold in your computer the same way.
Okay.
The advantage you have is just you have an easy interface to do with it, and also when you have a bunch of data, it helps you to kind of filter the information out. That's the part of it. So, like for instance, it plots things like this. You know, like that you need to analyze all the data. So, like this is here what you're doing, like all those structures. What you're trying to see, like, are them good? You know, and then when you look at this end here from 300 on, these are the results you have. And you see basically here, this is the score. 100 is perfect, 0 is horrible. So what we're trying to get is like how bad or how good is the structure and the composition. And that's all of them are bad in this area. So they're all kind of bad in the latch area. The reason for that is because the training set was not good at that. There's a lot of paper structures published, very few structures with the peptide. That actually shows one interesting thing that we actually made a bunch of studies on that, and we have this paper submitted and in bioarchive right now. That we compare AlphaFold with other systems. So we know, like in our science paper a few years ago, we noticed that every single one of these adhesins are very strong. So I basically told Priscilla, my postdoc, like if you get anything below 2,000, 3,000 like piconewtons, that's probably garbage. So she was trying many of these systems, and then what she got is like for AlphaFold, all these results here were coming. Like she was getting a structure that was giving her a decent result in terms of all the quality tests and all of that. But when she was pulling with SMD, they were relatively weak. So like many points here are replicas in the simulation. So these are crystal structures. See how the crystal structure is strong. All these were strong, but these ones are very weak. And then she tried the same thing with modeler. And then modeler was actually able to give her a better result than AlphaFold. So careful doubles. That's one of the main things. But another thing we noticed that I think it's more interesting than that, and that's what Priscilla doing because I would never notice that. That if you look at this and if you look at the bunch of these things at the bottom here, different strains of this bacteria, I would never notice that all of these are methicillin resistant. All the strong ones. And this one here is basically the same thing, methicillin susceptible. So this one is the bacteria, the older bacteria that we can barely find anymore because the one that likes antibiotics and dies from antibiotics, and the one that still survives antibiotics are these ones. So okay, that's weird. Is that a cool result that we found? So she started to look more details on that. She separated some of these. And for continuing because of many things, okay, so just one point, like why would we care about that? So like, first of all, resistant to most antibiotics, these resistant bacteria is a very important feature of them. They are what the CDC called the nightmare bacteria because, they are basically surviving our antibiotics. And they're the leading cause of death in healthcare facilities worldwide. So does this force play a role in methicillin resistance? So we started to look at the sequences for many of them. We picked, for many reasons, we picked some, right? Because you need to reduce your testing. We get this strain that we're calling S here, which is this has one of those complicated names. Okay, we're just calling S R1, R2, R3 to make our life easier here. So S is the susceptible one, and the resistant ones are R1, R2, R3. So the 3 resistant ones that we're testing. Okay? And if you look at them, there's almost no difference between them, and there's actually only one region, there's only one mutation of the peptide in this. And if you look at this, there's a serine there for the susceptible one, including in for the resistant one. Okay, let's simulate all of them more carefully, and this is what we get. Susceptible, resistant, they get stronger. Okay. We are testing that now experimentally. Lucas is doing that for us, not my son, the other one. And so just to give you an idea where that resin is right here, just that resin interface, it's right at the latch. It's not a direct read interaction with the peptide, but it's nearby. And this is the result we get, like one of the traces, like an exemplary trace. So see, like this is the susceptible one, this is the resistant one. And so Priscilla did something that was a cool idea from Lucas actually. You're like, can you try all the neutrons in that position? And then here it goes. The threonine is actually the strongest one. This is the susceptible one. The weakest one would have to have adhesin there. So like this is how basically changing. Like these are like each one of these points like 100 records. Okay? So like these are for you to get an idea about how that evolves over time. Like you can basically see that it's getting stronger and stronger. Okay, but here comes the question: methicillin does not act on.
Yeah.
So why would that be the case? The only reason we can come up with is a co-evolution process. Like if you survive longer, there's a better chance that there are going to be mutations inside that biofilm, that colony. So that's the only reason we could have. If you live longer because you are stronger in your side, there's a better chance that you're going to have more time to mutate, like in your colony, because you're reproducing that more chance. That's the only thing we could come up with. Yes.
So I noticed a very large variation of variances here. Is there any importance evolutionarily? Like, are some of these seen more that maybe have a more consistent strength instead of ones that, for example, there's one that dips all the way down to 2,500. Versus
those
on the plot that we were for this, yeah.
We don't know how much of that is artifact from the methodology. Remember, you're going away from crystals.
Oh,
back to this point, right? Like only a few of them are crystal. Like here, one of them is a crystal, the other one is not.
Yeah.
So it's can you actually trust? That's why we also do a lot of experiments. And actually, I'm not talking this part of the story here, but Priscilla is trying 3 different methodologies to get the same result.
okay.
So the idea was the following: if you have a crystal for one of them, and then you're going to continue like building on top of the crystal, you imagine that the crystal is going to influence a lot how the other one do, right?
Okay.
Even although you run long relation there. So what we've been doing is like we mutate from the crystal one, and then we get to the final one when you have a conformation that we say, well, that's stable for this other strain. Let's use that as a template now. We pretend the crystal does not exist, use that as a template to go back.
Yeah.
And we have been successful showing that we can actually see the same trend. The values change, but we see the same trend. So we see that the resistant ones are stronger than the susceptible ones, which is interesting.
Yes.
My last slide is just this. Okay, what we're learning from this. So first of all, I want to say, like, we are able to reproduce what they're doing in experiments really well. We have been able to do it by doing hundreds of replicas at different speeds. We can actually recover, use them to recover what they get in experiments, have an idea about what the force they're going to measure is. And we're being very successful there. By using these extra things like in analysis, like AI or any kind of other tools, that network is actually your thing we use every day. By using that, we have been able to actually understand better what's happening, to propose mutations and actually understand better the system. So I think that's very powerful. You can use that in a lot of stuff. And my main advice is like make a lot of practice. That makes a big difference when you're comparing that to experiments. And after that, I just want to thank a lot of people, you know, like from here, from everywhere that work in a lot of this system. This is my group back there. Marcel is not part of the group, he's not in the picture. But I also want to thank these three because I would not be anything without them. And these are the main authors of the things that I was showing here today. Like I said before, this is our department there, like our building. Thank NSF for the support. But you know, no one there cares about this building because everybody cares about this other one. That's what it's a football city. It's amazing. There's like when there's an important game, there's 200,000 people there. It's not a joke. There's 100,000 people outside the state, 100,000 people inside the state. It's insane. The department closes on Friday when there's a game on Saturday because you cannot walk. Thank you.
Very nice, very nice. I was really impressed, as you pointed out, actually, that we can really predict real things if we run it enough replicas. I was just lecturing about this morning, run as many times.
Yeah, we noticed that it's actually better to run more replicas than run longer solutions.
Right, absolutely. Very nice, very nice. Okay, so questions? Yes, please.
I have a question about the GPX box when you're running for it, because are you running it in big mode then, where you partition the GPUs into separate devices? And you're just in parallel, or you just use all of the GPUs out there?
It's all the GPUs and all the. At the end of the day, you notice to run it's a slower per record, but you run all 100 instructions.
Got it.
As long as you have memory.
Thank you.
This is Andy.
You had a question?
Yeah, the network analysis program, have you changed it much? Okay, so what are the major changes that you've made?
So we changed things to make it possible to run larger systems. That's one of the things we noticed. We changed the way you call. Because before you could basically do for one simulation when we're doing analyzing hundreds, we changed the way we call it. We didn't change anything in the main algorithm, like that. But like the way it's called, the way you assess memories and things like that, we changed all of that just because we're doing too much.
So can you do it over the, or you're saying you can do it over the multiple replicates now?
Yes, we do the so it's one shot. It's one call of the network analysis, and we do that for like 200 replicas.
And then just back quickly, so how many GPUs do you have on your VGX box?
So 24 in total. We have three with 8 each.
That's pretty good.
Yeah.
But you can also sort of program it differently, that you can run multiple jobs on one GPU.
Yes, so you can do that through Slurm. You can do that directly. Because I have a small group, we have Slurm installed. Never
Right.
For other users. Because like I have 3 postdocs. Each one of them has one. So you get your own GGX box basically. You can easily organize that.
That's what I learned actually from them. So you just have to load multiple jobs. You don't have to do a lot of things actually.
Yeah, if you are in a hurry for one specific system, we always like this machine is separated out for Priscilla, she's doing that, for Marcel, she's doing that. Otherwise, we keep putting it on. As long as we have memory.
That's not the advice that they gave us,
No.
right?
Because they want speed. They want to
Right.
show that your code is 10 times faster. I don't want to show my code 10 times faster. I want to show that I can use the machine as fast as it would be. So like I have a machine that's 1,200 nodes of blue waters. But if I have the basic like that's we target allocation,
Right.
direct allocation, then I know, but you get my point. You're fighting
I have a
for
blade of it in
allocation.
my office, actually.
Yeah, I have those.
Yeah.
So you're fighting for you're fighting those locations like all the time. And now it's there. I don't even apply for computer time because I want to apply for computer time when I go for much targets. At the moment, I don't need it.
That saves you time.
Rafael, you said that the old PDGA is actually connected, right? So
Yeah, they're interconnected, so you cannot actually use it the way you plan it. When we get to that point, it's gonna be better, but we're not there.
So this is with the MD2 as well,
We use them.
MD3. How do you apply forces? Is that allowed currently in MD3?
Yeah, now it's available for everybody. I have sent for more than
2 weeks ago, essentially, clean one is.
You had it early, I'm guessing?
So, yeah, we have an early, okay, the reason why I have an early version is because I pull in at 1 hour. Right, because I'm mimicking the experiment. And that's much easier to pull. So I have the version for it.
And you can still do these really fast polling rates with only one atom, like.
Yes,
Name, oh,
yeah, you're pulling only 1 hour. That's because they're mimicking the experiment, you're pulling the chain. That's, but like then we had to put another version, then we were the ones testing it because we do a lot of S&D. So that's
Sorry, I missed the beginning of your talk. I don't know.
okay.
So I was wondering about the CGS and this simulation that you ran. So what kind of force did you use Martin for that?
Yeah, Martini
And
and
like I'm imagining like you didn't use the net, was it called elastic network?
no, it should be the other one, the open.
Something like that.
Yeah.
But and also like I mean, it's very surprising because in Martini, like you have this restraint on the secondary structures and the forces that you're getting it matches very well with this.
Oh, it does. You missed one slide, and we got here exactly one slide late. So. So this is the result from Martini directly. But here's the trick. If you go to this pulling, the pulling velocity here is the same as this one, or this one, don't remember. So so now if you see this one here, the force is 3,500, the peak, in distribution. The distribution are basically the same shape. And the loading rate is 10 to 11. This one here is 10 to 10. And this is like you basically do a math there, simple math, and like you're going to see that you need to correct the loading rate by a factor of 8, the force by a factor of 2.05 kilogram. And if you multiply the two, all of them, multiply all of them by the same metric. So multiply all the points by the same one, the peak for the assume the slowest peak, our atom is their best result. Then you get it works for two different systems.
Do you know actually what in Martini causes this? So Martini, I remember they had a tendency to bring proteins together for the membrane proteins. For many years, actually, that has been a problem. And now you're saying that proteins don't interact strong enough.
You know how calcium is too strong for most of our simulations? For me, they're too weak. When you apply force on them, they're typically too weak. They're supposed to be stronger, like in many situations. So that's a problem of like pooling versus like thermal binding. So they're stronger for thermal binding. It might be the same case. It might be the same thing. It might be that the thermal inclination is, yeah, the barrier is smaller, but
Mariano, and then you go.
Yeah, my question is regarding like the first part of your work, the SMD part.
Yeah.
Do you think it would be possible to do like exchange Hamiltonian or stuff, or would you like disturb too much? And because that way you're not gonna replicate the experiments that you're gonna have from the equilibrium to the stretch. You may just like exchange different type of polynomials. Would you go over that or just keep the same way you're doing it right now?
okay, so a temperature one would not work. Because that's just a temperature one would not work because as you increase the temperature, you lower your barrier. So that would give you a wrong variant, so you take a benefit and that's exactly what you're getting. But then you would get, the problem that I see is that for each lot, you get a different loading rate. So this plot would become, I don't know, I think this plot would be a mess. You would not know how to fit the plot. Because here you fit, it's an annoying fit. You don't fit each one of the points. Basically, you fit the distribution on top of each one of the velocities, and then you fit again on top of the fit. So it's not a direct fit from the data. So.
Right.
Analysis?
Yeah, so you mentioned there was two different routes that can occur.
Yeah.
Was it fast route or slow route or high force? I forget.
Oh, yeah. So, yeah, one of them you're fluting with high force, it's high force one. The other one's a thermal filtration.
Do you see in coarse grain simulations versus the all-out simulations that there's one route that goes versus the other?
You didn't try it. You didn't try it in the course range at all. Because we were only doing the course range for trying to get a block like this. There's only one paper in the literature that gets which is connected. And was actually done the other way. So they use high-speed FM to get faster. They did the best they could with the Alada, but the Alada was kind of in this range here. And then the high-speed FM. That's a paper from Rico and that's the only paper that I'm aware of. So you're trying to get the same way. Can we bridge this gap with something that's cheaper? Well, that's kind of because to do this plot from blue here takes like a couple of weeks. To add this other two, it's not take as long. That's why.
So back on this plot, so if I recall, the polling, it's because of a system of hydrogen bonds, isn't it? The reason that it sticks so well. I'm not too shocked then that Martini doesn't get it because a lot of their things they need to do their elastic network to mimic those hydrogen bonds. So that,
I'm not surprised either.
okay, so I just wanted to verify that I'm not.
No.
Yeah. So you would need to do something.
To be fair, I was not expecting it would work at all.
Yeah.
But we're like, why not try it? Because it's relatively cheap to try one time.
Yeah.
And then, the only reason why this got so far is because Diego was like, I'm gonna try it again, I'm gonna try it again, I'm gonna try it again. And then he got to the point he was getting many plots. Then he noticed that, okay, he's getting the same profile, getting the same idea when it comes on one high speed, you have this curve. So can you actually find a multiplier here that would mimic the result? And that's not so important for this system. This system is good because it's all behaved. So we decided to do it. But actually, he started doing that because he's working on antibodies together with Mechanoactive. And antibodies are very small molecules, low force, and that's a very complicated system. And there, these simulations are going to be helpful for us.
The same line. Hydrogen within the program, between the program. Suffer from the same. Yeah.
Right, exactly. Yeah. The first time he tried it, everything was forward because the way, you should turn off and the other one.
And that's probably why you need the same scaling factor. For the protein, the number of hydrogen bonds is different.
Yeah.
Very good. There's a reason if you look at the maybe the free energy expression that you get about the same parts of it, maybe at the end of the day, it'll work out. Or send it to her. She wants to seriously. She might be releasing the side. She looks at her.
I don't know.
I think she'll be the before.
The deal is going to be there. She works on these also, but the same kind of idea before. Then look for like the catch bond behavior that we're actually doing from this deal.
Yeah. Any other questions? Comments? Well, thank you very much, Rafael.